stem cell conversation held on behalf of ASSCR. So there's been a slight change of plans. So today we'll be in conversation with Janet Rossant, but also Christine Wells, our very own Christine Wells. Nathan wasn't able to make it today. He's had a bit of a family emergency. Not to worry, we will soldier on, and I'm sure we'll provide a very compelling conversation for you. So just another bit of news here. Today is being recorded. There are no data presentations today, but just to note that, should you wish to participate, today is being recorded. So please keep that in mind. There's another exciting feature that we're introducing today for the first time in the Stem Cell Conversations, which is the inclusion of this Slido app. So this might be new for some of you. There is a little explanation in the meeting chat at the bottom. There's a link there that you can follow. Otherwise, you can scan the QR code that's on the share screen with your phone. You can either view this on your phone or on your computer browser. Either will be fine. And I'll be encouraging people to sign into that. Or you should just should take you straight to this page where you can start, or some people already have. You can start inputting your questions for the panelists. And if you like any of the questions there, you can use the little thumbs up. And the more likes a question you get, the higher up the list it will go, and it will be pushed to the top of our panelists' attention. I'll get back to basics though. So welcome to today's Stem Cell Conversation. My name's Jarmon, for those of you who don't know me, and I'll be your moderator for today. We'll begin though by acknowledging the traditional custodians of the land on which I meet you from today, which is the lands of the Wurundjeri people of the Kulin Nation, and we pay our respects to their elders past, present, and emerging. And we might also stop to consider how our research might affect the lives of our First Nations peoples here in Australia. Does our research contribute to closing the gap? And if it doesn't, is there a way in which it could? Are there opportunities for us to engage with our First Nations peoples as a consumer group when we're writing our grants? And if that's something that we could do we know how to go about doing that as well? And do we know who to reach out to start that process? And there are people out there that you can reach out to. So if you don't know, you can contact me, but you can contact probably Christine as well. So as I already mentioned, today is a little bit different. We're going to be having a conversation with Christine and Janet on all things sort of journals, peer review, the editorial process, and of course stem cell reports as well, seeing as we have the editor-in-chief of Stem Cell Reports with us today. So I've already sort of covered, there are more people coming in as I'm speaking. So I've already covered there are a couple of different ways in which you can participate today. You can either use the raise hand function in Zoom, which will take you to the top of my screen, and I'll be able to acknowledge you and ask you to ask your question directly, or you can use the Slido app. So you can scan that QR code, or there should be a link in the meeting chat, which will take you to that on your browser as well. So feel free to start adding questions to that app. And or liking the questions that you would like to ask the panelists. So I'll start by briefly asking Janet and Christine to introduce themselves. We'll start with Janet, who is the president of the Gardner Foundation and senior scientist emeritus at the Hospital for Sick Children in Toronto, and very relevant today, the editor-in-chief of Stem Cell Reports. So Janet, would you like to briefly walk us through your journey so far, touching on how you and why you became the editor-in-chief? Chose to become editor-in-chief at Stem Cell Reports. Thank you.
Okay, well, thanks, Jarmon. Now, I've had a long career in developmental biology and stem cell research, and that's really my major interests. Along the way, I've done editorial work. I was an editor for a time at Development, which is a very well-known development journal. I've also been an editor for eLife when it started. I've been an editor for PNAS. I've been on the review board for Science. So I've had a lot of different editorial experiences. But why did I take on this one, which is not just being one of the editors, but being editor-in-chief? Well, actually, it's because I actually needed something to keep my brain working. So I have now emeritus, as you just said. I closed my lab, which I've been running my first paper was published 50 years ago. 50 years ago, a scary thing. I closed my lab a couple of years ago, but I'm still very much engaged with the stem cell community, with ISSCR. I'm very much engaged in the developmental biology community with SDB. And the opportunity came up to take over as editor-in-chief of Stem Cell Reports. And I thought that, this is a very good way to contribute back to the community, because Stem Cell Reports is a not-for-profit journal. It's the ISSCR's own journal. So ISSCR is the backer of Stem Cell Reports. It's published by Cell Press, but it is not a Cell Press journal. It is not one that makes money for Cell Press. If it makes any money at all, and it doesn't make much, that money goes to ISSCR and goes back into promoting things that we as stem cell scientists like to do. So I like the concept of it. It's an open access journal. That's very important. And as I say, it was a great opportunity to stay connected with the stem cell science and be able to look back over all the things I've learned over the years and my current experience in science to look across the board at what stem cell science is today, from the fundamental science through preclinical to clinical applications and to ethical and regulatory issues. And that's what Stem Cell Reports covers. And that's, to me, something that I don't know all about all those areas, but I know enough about all of them that I thought I could be a useful editor-in-chief. It would keep my brain going, but it would also, my experience is such that I think I could lend some expertise to the journal. So I was asked if I would be interested. And in January, I joined as editor-in-chief, taking over from Christine Mummery. Christine Mummery was the first editor-in-chief of the journal, taking over then from Martin Perrow, who was the second editor-in-chief. And so I have two great scientists ahead of me, and it's up to me now to make sure that we keep up the reputation of the journal. And the journal really is a rigorous journal run by scientists for scientists and represents some of the best research that's going on in stem cell research today.
Thank you, Janet. Yeah, no, that's fantastic. We're going to come back to a lot of the sort of points you covered, in particular, sort of to what extent being the editor-in-chief occupies your time, because I assume it's not an insubstantial sort of role, and also the direction that you're hoping to sort of take Stem Cell Reports into as well. We might just hear briefly from Christine and sort of your journey in the editorial space as well, and then we'll start opening up the floor to questions. Thank you, Janet.
First of all,
Thank you, Christine.
apologies to those of you who are expecting to see Nathan in this seat. I know I'm a poor substitute, but I'm certainly very happy to chat to you about my editorial experiences. So I'm Professor of Stem Cell Systems here at the University of Melbourne, and I'm a computational stem cell biologist, which means I use computational modelling to understand and predict cell behaviour. And then we use pluripotent stem cells in the lab to test those models. I've been working as an editor in various journals, mainly genomics and computational journals, for the last 10 years. And I got into it actually by asking a mentor to mentor me into first a reviewer role and then an editor role when I first set up my own laboratory. So I guess there's often questions about how do you get into this, and sometimes it's just by putting your hand up. I love editing. I'm currently an editor at the journal PLOS Genetics, which has got a very broad focus on inherited and genomic technologies and applications. But I also have a link to Stem Cell Reports. I was the first member of the editorial board to introduce computational stem cell biology as a theme within the journal. And that's now ably covered by Patrick Cahan on the editorial board. Why do I do it? I think because you are taken out of your own little area of expertise and interest, and you get to see a breadth of research and a breadth of approaches and a breadth of perspectives that you wouldn't see otherwise. And that is, very exciting and engaging and a wonderful thing to be able to benefit from, really.
Thank you, Christine. So I apologize. I did have actually a spiel to introduce you, which I forgot. But then again, Christine is extremely well known to this community. So in a way, you need no introduction. But my apologies for that. So we'll now turn to our sort of interactive part of this, which will cover the rest of the session. So we're now going to start asking our panelists some questions. So anything you've ever wondered about peer review or becoming an editor, or what's involved in being an editor of a journal, and what you think about, well, not what you think about stem cell reports, but what's happening at stem cell reports, only good things, of course, and so on and so forth. So we have that Slido app. Please start putting your questions in and voting for the questions you like. Otherwise, use the raise hand function. I might kick things off though by a lot of our community, or a lot of the people that chimed into these conversations are early career researchers. Could I ask Janet, perhaps, if there are any sort of stem cell reports features that are specifically targeted towards early career researchers, in either in the peer review or in the editorial sort of space?
Yeah, well, stem cell reports actually has quite a unique, never say that because somebody will say, no, somebody else does it. We have a very special engagement with early career investigators, which is the early career editorial board. And as you may have seen and probably know, because Nathan Palpant is one of the members of that board, we actually have a special group of early career investigators, people in the sort of the first 7 years of their career as an independent investigator, as essentially editors for the journal. We call it the Early Career Editorial Board, but actually we pair up those junior researchers with a senior editor, and they go through the whole process. They actually handle papers, and they look at the reviews and they recommend a decision. So the whole process, they learn how to do that. So it's a fully mentored program. So that's really quite special. It turns over. So we've just finished. So it's something that Martin Perra brought in his term of office. And so we've just turned over the first ones. So the people who came in have been dropping off. And we've just we had a competition, an open competition for members of ISSCR. So you have to be a member of ISSCR to potentially be early career editors. And we had a lot of interest, and we've just appointed 3 new early career editors who will do it for a sort of 3-year term. So this is a rotating opportunity for early career people to join. So that's one important thing. Somebody else is asking, I'm running ahead here of the questions, but one of the questions is, how do you get to be a reviewer at a journal like Stem Cell Reports? Well, one of the ways you can do it, there is on our website, you can sign up and say, I'm interested in being a reviewer. And we will certainly look at those applications. And if we think, you have the capacity and the expertise, then we'll put you in our pool, and you may get called on for a review. So that's one way. We can talk about other ways, but that's something that Stem Cell Reports specifically does.
Thank you. Yeah, obviously that might be a unique aspect of stem cell reports. It might not be, but other journals have peer review programs as well. So obviously the editorial thing is probably for more relevant to the more senior people in the audience at the moment. But for early career researchers, sort of the PhD or very early sort of postdocs, being involved in a regimented sort of peer review process as well is something that I know that some other journals do. Is that something that Stem Cell Reports is interested in doing as well, pairing up early career researchers with senior editors to do regimented peer review and get feedback on that?
We don't do that specifically. We do encourage PIs, if we ask a PI or an early, at whatever stage in their career, to undertake a review, we encourage them to share that process with senior postdocs or students, as long as they tell us who they're doing. And then those people will also get recognized in our list of reviewers for the year. So they will get recognition for that. But we encourage it to go through the PIs. We don't, at the moment, have a specific program for training peer reviewers. Cell Press has a lot of material, however, online that when we encourage people to do reviews, we do, because we're a Cell Press journal or published by Cell Press. There's a lot of information that Cell Press provides to try and to provide the outlines of how to do peer review and how best to go through that process. So there's a lot of online material that they can access.
Now, I've clocked you, Lincoln, but I might just jump in with one more question before we come to Lincoln. You mentioned that, yeah, your peer review activity will get recognised by stem cell reports, and I believe this goes for most journals as well. Would you just like to sort of expand on what that means as a peer reviewer? If I'm a junior peer reviewer, or any peer reviewer really, what sort of information might get collected by the journal and used in what way?
Well, at the moment, that's a good question. I can't give you absolute chapter or verse. I know that we list all reviewers at the end of the year, so that's something that we can give you, that we have that list. And also, if you're a peer reviewer and you reach out to us and say, could I get a letter from you to say I did this peer review, then we will do that as well.
Because I think it was quite surprising to me to realize that actually my peer review was being ranked and that I was being assessed by the handling editor at the time. I didn't quite realize that. It made me get my reviews back in a more timely fashion when I realized that.
We do that as well. It's true.
Lincoln, do you want to jump in? Oh,
Lincoln.
you're still muted,
Lincoln.
unfortunately.
Yes. All right. Where has Lincoln gone?
I think it's a technical issue because you are unmuted. You might have to type it into the Slido, Lincoln. Sorry.
Can I add something, Jarmon, to that question of how do I get on board as a reviewer? And it relates to your second question. I'm asked by my PI to review manuscripts. Can I be credited? I would very much encourage anyone who is being asked to review a manuscript to actually negotiate that credit up front with their PI. It's really important that the journal knows who's reviewed the paper.
Absolutely.
Because scientists reviewing scientists, you want to make sure that it's being reviewed by people who've got the experience and the expertise to really look at the manuscript carefully. But also, I do this myself. You want to train your postdocs in that peer review process. I think it gets you to write better papers too. So the easiest way for the PI to let the journal know is simply just to write to the editor and say, I'm happy to take this on, but I'm going to ask so-and-so to do this. And then you're in the system. And you're more likely to be asked again to do it if the quality of that review is good, because when behind the scenes in the editorial, in the paper handling system, you can actually see how often someone's been asked to review a paper, how well they turn it around, and so that can help you, I guess, weight whether you're going to ask someone over another person. If they rarely say yes, or they're often very late, you might not go to them first, for example.
And I would just comment, of course, which I think every editor would know, that junior people who do reviews tend to be much more rigorous in their review, sometimes to the extreme. So a message also back to reviewers. So if your PI hands it over to you and you get to review it, just remember how the first paper that you wrote as a junior PI or a junior investigator, how upset you were when the reviewers really ripped your paper apart, and try not to just do that in return. So peer review is really to provide a critical but constructive critique for a paper, looking obviously if there are real issues that are of data that is incomplete, data that needs revising, but try not to go overboard, because that is the other thing that will not certainly won't endear you to the people you're reviewing, and probably in the long run not to the editor either. So there is a balance to be had there. So I do encourage when PIs get their postdocs involved, that the PI must also have a look at that review, just to make sure that it is balanced and it's not too critical. We sit in journal clubs, right? And you rip a paper to pieces. We can all do that. That's not really what we're looking for. Particularly at stem cell reports, what we're asking is this paper present rigorous research. So we really want you to look at the data, the methods, the approaches. Show us that this is providing rigorous evidence for whatever it is that they're trying to claim. And that's really what we're after. That's where you can put the time and effort. If they overinterpret their data, you can say they're overinterpreting, but just try to keep it on an even keel.
Do you have a rule of thumb, either of you, for that sort of thing? Is it like 2 or 3 main concerns within a paper, but any more than that, and you might sort of say, all right, we'll move it on? Or
well, you mean when it comes back?
Yeah.
No, that's really hard. Thank Kristen's shaking her head too.
Yeah.
It's very hard to come up with a hard and fast rule. There's also, remember, we always get more than one review. So certainly if 2 or 3 reviewers have serious concerns, especially if they're the same concerns, then that kind of makes you feel that maybe this paper needs to go back and be rethought. We do reject, but we tend to we have something in stem cell reports which we call something like provisional rejection, which not quite, I was quite surprised about that, but it's kind of quite nice, really. We can't take it right now, but, take all these comments, and if you come back and really bring us the data we want to see, then we'll look at it again, because we're here to help. We're not here to take you down. We're here to help all researchers provide better papers, provide their contributions in the best way to the community. So it's very rare that we if something's gone out for peer review, it's very rare that we will totally reject it out of hand.
And I guess that comes to the top question there on the poll at the moment, which is, what are the key points that you as editors and Janet's an editor-in-chief look for when deciding whether to actually make that first commitment to the paper to send it out to review?
Yes.
People are obviously looking for a way in here. So what can you offer?
Yeah, so this is I would say the trickiest one. So stem cell reports, I understand that stem cell reports is not Cell, Nature, or Science. We don't intend to be Cell, Nature, or Science. We're not in the competition for the latest, greatest, story. We're here to look for really important, rigorous contributions to stem cell science. However, we do not send every paper out for review. And that does come down to the fact that we receive a wide range of papers, many of which really just do not reach that level of rigor that I was mentioning. So we get a lot of, if a paper does not, maybe it's easier for me to say what a paper should have to be sent out for review than what's wrong with the papers that we don't send out. So the key, which is really the question that was asked, what are the key points we're looking for? We want to see that there's a clear hypothesis that this is addressing a specific issue in stem cell science. It could be from fundamental stem cell biology and model organisms right through to a preclinical study or even a clinical study on something that is really showing the impact of stem cell biology on clinical science. So it can go from basic biology to mechanisms of differentiation, but it has to be addressing a specific question. There have to be clear, rigorous experimental protocols presented, and there has to be a conclusion that looks as though it's moving the field forward in a way that the broad stem cell community would be interested in reading this paper. So if it's too limited in its advance, we will not be interested. If the data is not rigorous enough, if the experimental approach is not rigorous enough, or if it's really repetitive of many papers in the field, then we're probably not going to accept it, send it out for review. So make sure your paper has really rigorous approaches, has a clear rationale, and is making a contribution that if you were reading it, you say, this is something I'm glad to see in stem cell reports. So again, it's hard to be absolutely clear. I can't tell you, I can't show you the papers we accept and the papers we don't send out, but I can tell you that the papers we send out represent what we hope to see as major contributions, and are the ones that I would like to apply the things I just said, that we'd like to be working with those authors to improve their paper and really have it presented to the world.
I think it's worthwhile thinking about what the different cover letter, abstract and full paper, how they're looked at in the editorial team as well. So a good cover letter will really summarise the key highlights very succinctly. Even dot points are great. They'll say to the editors, this is my audience, and this is where I'm positioned within the field. So where this is the question we're addressing, and we're addressing it in these ways. It will also include some really critical information, for example, that all of the authors have agreed for the submission, if data is being embargoed until publication, how the reviewers can get access to the data, but really providing a very clear summary of why the editors should look further. An abstract, that is what gets indexed in PubMed, and it's often the first port of call for a student to browse through before they go into the paper proper. But jumping in and having a look at the quality of the figures, quality of the data, and the quality of the methods that are used, and the quality of interpretation of those is really what I would do as an editor before sending it out to review. If the grammar is poor, if the manuscript's not readable, then it wouldn't go out.
Yeah. So the question said, do we look at all of it? And Christine is quite right. We make that decision on looking at the full paper. We don't make the decision simply on the cover letter or the abstract, or those. They can be helpful, but when we're making that decision, it's on the full paper.
Yeah. It's a very collaborative process as well. You have an editorial board, and there are mechanisms within the editorial manager to reach out and ask questions of other editors. So there's often a conversation about papers that you're not sure about before it's sent out to review. There can also be a conversation about who would be suitable to review it. So this is not a sole person making a sole arbitrary decision. It is a very collaborative process.
We're gonna quickly move on because there are so many questions being asked and so little time. 2 minutes left. This one I think is quite dear to your heart, Christine, and I think we've mentioned this before as well. In what way is AI being sort of monitored or allowed to be used, or in which way, in how can you even know that it's being used in peer review? What's STEMSEL Report's policy on this, and where do you see that going into the future as well? That's a lot to tackle, but your best guesses, Janet, Christine. Thank you.
I'll let Christine take that one.
Look, this is really hard, right? Because the truth is that these generative AI tools are really accelerating very quickly, and they're being used much more widely than I think any of us can really appreciate. So obviously there are plagiarism tools which journals will use that can help you identify likely AI generated text. And we do ask people to declare if AI has been used. I think in terms of, is it okay to use AI in image preparation? Well, some machine learning tools would be classified as AI, and they're a core part of the analysis pipeline. So in that case, the question is clearly yes. If you're asking AI to generate the figure and it's a schematic, maybe that's okay too. But if you're asking them to generate primary data, I think that obviously crosses a line. So at the moment, I think the honest answer is we are expecting that authors will declare when they're using AI. And in the meantime, we are working very hard to get better at detecting inappropriate AI use. Janet, how does that fit with the STEMSEL Report's experience at the moment?
Yeah, I think that's right. Cell Press has some actually is very tough on AI. and really does not allow AI to be used in writing the papers, manipulating the images, or as we just found out this month, in making a pretty image for the cover of the journal. You're not allowed to use the AI, which I find a little odd. Because it's just it's just a diagram. Why not use AI? Most tools essentially use AI. So I think this is evolving. If you are using AI, and for a lot of people, especially if English isn't their first language, the generative AI tools can be very useful, but you have to acknowledge that as you write the paper, and you have to say how it was used and what it was used for. So it can't be used to generate the paper or generate data, but it can be used to help edit and improve the language of the paper. But it's going to evolve. This is an evolving, a totally evolving area. I don't know where it's going to end up.
Thank you. There's a slide on at the moment, which is, and I apologize, it's giving the answers in percentages, which I don't think is actually very helpful. And people can answer for more than one question, which is why the percentages don't add up to 100. There are a few issues there, but I think just generally, there are about 15 people who have answered, so that'll give you some idea there. But this is just a bit of a view of that people are interested in getting more involved in peer review. People are interested in getting more involved as an editor. You've already briefly touched on this, Janet and Christine as well, but again, could we just revisit how people might be able, if they have absolutely no idea whatsoever, how would I get involved in peer review? Let's start with that one, please.
Well, I would start by asking your PI, asking your group leader, can you support me do this? So when they are asked by the journals, they may not have considered that this is something you would want to do, and it might be something that you do together. And the way I do it with my postdocs is I give them the paper to review, I review it, and then we get together and we compare our reviews and just talk through the different approaches. Particularly, as Janet's already mentioned, our job as reviewers is not to redesign the experiment. It's not to suggest, the upgraded model of experiments. It's actually to assess whether the claims made in the paper are supported by the data that they have.
Yeah, absolutely agree.
And Jen, I think you mentioned that there's actually there's a section on the Stem Cell Reports website where you can sign up, expression of interest, basically, to be a peer reviewer. Was that?
There is actually,
there is, yeah. So there you go. In terms of getting involved as an editor, that one's a little bit more complicated, isn't it? That might involve an invitation from a journal, which will probably be based on your ongoing and sort of contribution to the field, which I think will be more relevant possibly to the senior people. Is that?
Yeah, and there's two levels of involvement at the editor level. You can be on the editorial board of a journal, and that's a larger group of people who can be called on, as Christine said earlier, for advice. They can be called on to review. We have a new rule at Stem Cell Reports that says if you're on the editorial board, you must agree to, if asked, to review X number of papers per year. Most editorial boards, I discover, don't do that. And it's like, wait a minute, shouldn't you be asked to do some reviews, do some work for the journal if you're going to sit on the editorial board? So that group still tends to be more senior people, but it is one where, as a up and coming scientist in the field, you may find yourself having the opportunity to join the editorial board, and it doesn't hurt to ask. If you're engaged, particularly something like a society journal, if you're engaged with the society in various ways, you can ask whether you could be considered for the editorial board. The editors, and there's usually only a smaller number, that's a more complex process, because a journal is usually looking for editors to cover specific areas, specific geographies, specific seniorities. And for ISSCR, because it's a society journal, there's actually a society committee that looks at the composition of the editorial board and the editors. And there are open calls. I told you there was an open call for the early career editorial board, and there can be open calls for editors as well. So it's worth watching out for those.
Can I just ask the final question? I'm going to be cheeky and ask the final question. And this one was a gift from Christine earlier, actually. And this will apply to both of you for your respective journals. what's going to, or what do you pitch? And obviously as editor-in-chief, you are uniquely placed to comment on this. What's going to be hot in 2026, and what is Stem Cell Reports going to get really excited about in 2026, and then Christine as well, in terms of PLOS Genetics.
Well, okay, that's an interesting question, because certainly for Stem Cell Reports, we really are seeing in the stem cell field, we're seeing that the science is moving closer to the clinic. I would like to see more preclinical and clinical studies in Stem Cell Reports. We tend to be still at the fundamental end of stem cell science, which is, where I belong, I guess. But I see the field moving forward. I see the applications of computational biology, of tissue engineering, of all the subsidiary science and AI, the subsidiary sciences impacting on stem cell. And I want to see more of those interdisciplinary studies coming forward. And I expect to see that in the next couple of years.
Thank you. Christine?
I think in PLOS Genetics, the trends are actually new technologies that are giving us a different lens onto the activity of the genome. So, single cell technologies and spatial technologies really allowed us to understand how gene expression was coordinating the behavior of individual cells. Now, I think we are starting to see long-read technologies which let us look at the particular gene products that are being made by different cell types, and the diversity of the transcriptome and the proteome is really remarkable. We're also starting to see much more combinatorial measurements of genome structure and genome output, which is exciting because we're moving from what's essentially very descriptive science into starting to build mechanisms and rules. In the stem cell space, I think stem cells are an excellent model to be exploring some of these things, because you can get these transition states at scale in a way that you couldn't do in an animal model or a human. So I hope that we're continuing to get deep insight into our biology, what makes a cell, how cells engage with each other and their environment through these different technology lenses.
There's
Thank
no
you.
question that in the stem cell space as well, obviously single cell analysis, spatial transcriptomics, spatial metabolomics, spatial proteomics, the tools that we have to explore every aspect of biology impact clearly stem cell biology as well. And that will continue to be a focus for stem cell reports. But as I say, we are about stem cells. We should be thinking about moving them to the clinic. And of course, even PLOS Genetics, of course, genetics is having an incredible impact in the clinical arena as well. We're seeing how understanding genetic pathways is beginning to really translate into personalized medicine. So it's an amazing time.
Absolutely.
There are some lovely formalized programs, but obviously it's journal specific, run by specific journals that will bring ECRs, students and early career researchers in as a peer reviewer. They'll pair you up with a senior person. I've had hit and miss experiences myself with that. You can get bombarded with too many or none, and I never really received any feedback on my actual peer review either. So anyway, we should probably stop the recording at this point, but before I get myself in trouble. But then I think probably the most eye-opening experience I've had so far is being a guest editor for a journal and seeing what goes on behind the scenes there, and getting the feedback back and trying to work out what to do when I got a reject and accept and a major revision, and I'm like, I have no idea. I pushed it up the chain of command. I was like, I'm out of my depth here.
That's exactly the right thing to do. These are really collaborative processes. And that's the great thing about a reputable journal, society journal, is that, they've handpicked great editors and they will draw on their editorial board for those discussions. Company of Biologists has got some great opportunities to get experience with peer review as well. They've got the pre-lights activities where you can peer review bioarchive papers and have your review published on the pre-lights website, which I think is really nice. It's like a journal club, I guess, for PhD and postdocs. Some journals, like the PLOS journals, will let you see what the other reviewers said, actually. So
Yes,
there can
yeah.
be some collaboration between the reviewers as well.
But thank you very much, Janet, for your time. Thank you.
Okay, thank you. Nice. Thanks, Christine.
Thank you.
Thanks, Jarmon.